In this video, we're going to take a look at the vessels themselves and discuss the changes that the vessels undergo as they get smaller and smaller, heading away from the heart, and then larger and larger as they go back toward the heart. And we'll look a little bit about the exchange that occurs at the capillaries, so how we actually get oxygen and nutrients and hormones and all those things out into the body, and how we pick up the waste materials at the far side of the capillary network. So let's get started. Arteries and arterioles conduct blood away from the heart, and they are composed of all three tissue layers. The arteries are, I'm sorry, not arterioles, but arteries have all three of those tissue layers that we discussed in the last video. Excuse me. The tunica media is the largest portion of the artery wall. It's relatively thick, and it's there, as we've said before, to withstand the high blood pressure that can develop or be developed in the arterial network, or the arterial vessels. So large arterioles also have all three of these layers, the tunica externa, tunica media, and tunica interna. But the farther away from the heart, the less and less thick the tunica media becomes. And when we get down to the small arterioles, they may only be composed of the tunica interna, that's that lining, the endothelium, and a few scattered smooth muscle fibers, like we see here in this cartoon, and have almost no tunica externa around them. So as we get to the smallest blood vessels, we're losing tunica externa and tunica media. So that by the time we get to capillaries, which are the smallest of the blood vessels in the body, by the time we reach them, they are composed only of tunica interna. So now we've lost all of the material that helps to prevent the movement of any materials out of the blood vessel. They're found in these branched, interconnected networks. So if here's a small arteriole leading into, so this is the arteriole coming into the network, and so there are these cross connections so that blood can flow in multiple directions through the capillary network. The density of how many networks are found in a particular tissue is all related to how metabolically active that tissue is. So if it has a lot of metabolic activity, let's think muscle or the liver or the brain, then there's going to be a high density of these capillary networks out there. While if it has a low metabolic rate, I'm trying to think of something that might have a low metabolic rate. Some organs, some of the organs of the abdominal pelvic cavity might have lower metabolic rates. They're going to have less of this blood supply available to them. Cartilage is another good example. Look, it's right there on the slide. And there are some tissues that have no capillary networks at all. Think here, for instance, the tissues of the cornea, the clear covering at the very anterior most surface of the eye that we look through. If there were capillary networks there, then of course it would be blocking the view of the world beyond us. So these capillaries also have relatively large gaps between the cells that make up that endothelial lining. They're referred to as intercellular, so between the cells, channels. So there's these relatively large intercellular channels, and this allows the exchange, and so that's what they're trying to show here, both ways, exchange of materials into and out of the blood and into and out of the interstitial fluid that surrounds the capillary. And the size of the channel, that intercellular channel, is what controls how permeable the capillary is. So these intercellular channels are larger near endocrine glands where we're trying to pick up hormones, and so that will allow more of that hormone to be transferred into the blood. They're large around the small intestine where we're trying to pick up lots and lots of nutrients there. And they're large at the kidneys where we're trying to squeeze out of our blood all the fluids so that we can then only selectively call back into the blood the bits of the blood fluids that we want to keep in the body. So size of the intercellular channel is what controls the permeability and the amount of exchange between the blood and the interstitial fluids. So the entrance to each capillary network is controlled by a little band of circular smooth muscle. So this is just a couple of muscle fibers that wrap around the capillary network entrance and can control whether or not blood flows into that capillary network. The band of smooth muscle is called a pre-capillary sphincter. This is the first time I think that we've encountered this term sphincter. A sphincter is just a band of muscle that wraps around a tube, and it can constrict. The band of muscle can, and that will close off the tube, or it can relax, and that opens the tube back up again. And this precapillary sphincter monitors and notices the concentrations of oxygen and the concentrations of nutrients that are here in the tissues, so in the fluids here around the capillary network. And as it monitors, if it notices that the blood, the oxygen supply in those tissues has become low, then the precapillary sphincter will relax, and that allows blood into that particular capillary network. And then as oxygen levels rise again, the precapillary sphincter notices that, and it will constrict, and that shunts the blood then off to other capillary networks that are in tissues that need more oxygen. So we don't have all of our capillary networks open at the same time. They open only when they need oxygen or nutrients, and then they close down again so that we can shuttle the blood to the places where it needs to be. So there are two things then that control how we route blood flow through the body, and it's the vasoconstriction and dilation of arteries and arterioles that either allows blood to flow freely through the artery or the arteriole, or they close down a little bit. The arteries and arterioles don't shut off completely, but they can close down so that more of the blood gets moved to other more open arteries, and it's controlled by the contraction and relaxation of these precapillary sphincters. So we got a fair bit of control over where we move the blood to, and it is primarily determined by the needs of different locations of the body. If they need oxygen or nutrients, then the vessels will dilate, and if they are good for oxygen and nutrients, then vessels will constrict. Okay, so now we get to the piece that's about how we exchange materials in and out of the capillaries. And it occurs by processes that we've discussed a lot, but it was a long time ago, last semester, that we looked at it. So diffusion is one means of moving materials, and when we diffuse things, we move them along their concentration gradients. So I want you to think now, and you may want to pause the video after I ask the question. Think about what is the direction in which materials diffuse. And so pause now, and then you can come back and we'll answer. Okay, hopefully you thought about it some, and you remembered that materials diffuse from high concentration to low concentration. So in the systemic circulation, where the blood that enters the capillaries has lots of oxygen in it, and may have lots of nutrients as well. Diffusion will move these materials out of the blood in the capillaries where oxygen is high and into the interstitial fluids where oxygen is lower. So we move materials from the blood into the tissues. And it all can happen just by diffusion if they are materials that can make it across the, out of the blood vessel. And remember now, we've seen that these intercellular channels are going to allow a lot of fluid to escape from the capillary network. So out there in the systemic circulation as well, around those capillaries, concentrations of carbon dioxide and of waste materials are going to be higher. And so those materials are going to diffuse from the tissue fluids into the blood. So diffusion moves materials from where they are in high concentration to where they are in lower concentration. Filtration also occurs, and this is going to be about those intercellular channels too. So blood pressure can provide a source or an impetus for filtration to occur. And so blood pressure, where it is higher, will push fluids through those intercellular channels, and we filter out of those fluids or they remain in the blood, I should say. Okay, so the large molecules that are found in the blood remain in the blood because they don't fit out of the intercellular channels. So plasma proteins are going to remain in the blood because they cannot go out through the intercellular channels. And then last of all, osmosis. Here's where you need to think again. Be ready to pause. What material is moving when we use the term osmosis? So take a pause and then check back in. Okay, hopefully you thought about it and came up with the idea that osmosis is the diffusion of water from regions where water is in high concentration to regions where water is in low concentration. And so those plasma proteins that don't get squeezed out through the intercellular channels, they displace some of the water, making water concentrations low in the capillary network once we've squeezed all that fluid out. And so the colloid osmotic pressure of those plasma proteins draws interstitial fluid from the tissues back into the blood. Again, down that concentration gradient for the water. So three means of moving materials back into the blood or across the blood vessel walls. Diffusion from high concentration to low concentration. Filtration, where we push a fluid through a small hole and trap larger molecules on one side of the hole. And osmosis, where we draw water from a region of high water concentration to a region of low water concentration. Okay, so here, this image is trying to make sense of this. So here's blood coming in from an arteriole. This is blood and we're out in the systemic circulation, so this blood is high in oxygen. Now, at the beginning of the capillary network, we have fairly high blood pressure, about 35 millimeters mercury. Now, compared to the aorta, this is low blood pressure, but on the whole, though, this is a relatively high blood pressure compared to what else is going on. Now, that's a force that's pushing out. Now, there's also an inward call based on colloid osmotic pressure. That has a net call inward of about 24 millimeters mercury. So if we have a 35 millimeter push out and a 24 millimeter mercury push or call in, if we subtract those, there is a net call of about 11 millimeters mercury. And the direction of it is, there's a net pressure of about 11 millimeters mercury, and the direction of it is going to be out because that was our larger value. So we have this net outward push of 11 millimeters mercury. So filtration is high here at the arterial end of the capillary network. We push fluid out, and that fluid, as it goes out, is carrying with it oxygen, hormones, nutrients, anything that is small and can squeeze out through those intercellular channels. Now, blood pressure drops across the capillary network, because of some forces that we'll look at in one of the next lectures. We see a real drop in blood pressure. So by the time we get to the far side of the capillary network, over here near the venule, the blood pressure has dropped to about 16 millimeters of mercury. And there hasn't been a lot of change in the colloid osmotic pressure. So now we have 16 millimeters mercury out and 24 millimeters mercury in. So we do the same calculation, and if you do the math correctly, you should find that there's 8 millimeters mercury pressure, Hg, there we are, that is this time negative. So that means that if positive is outflow, then negative in this case is inflow. So here on the far side of the capillary network, because hydrostatic pressure, the blood pressure, has decreased a significant amount, we see that there is an inward call for fluids, for the water, the interstitial fluid. And it brings in with it carbon dioxide that's out here in the tissues. It brings in with it any hormones that maybe are being produced out here. It brings with it waste materials from metabolic processes. So we see a flow outward followed by a flow inward. Out, carrying all the goods out. In, bringing all the goods in. So one last thing to say about this is that the call inward is not as strong as the push outward. So this leaves more fluid out here in the interstitial fluid than, every time blood goes through a capillary network, we don't call all of the fluid back into the capillary at the far end. And we'll see in the next chapter that that is an important function then of these lymphatic capillaries, is to capture that extra fluid and return it to the blood. Okay, so exchange in the capillaries is moderated here by hydrostatic pressure in a big way, by colloid osmotic pressure, and then of course we also have a lot of diffusion happening that is pushing oxygen from high concentration to low concentration, pushing it out, and drawing in carbon dioxide from high CO2 concentrations to low CO2 concentrations. Okay, hopefully that made some good sense to you. Got my fingers crossed. So, and here I am just saying the last little piece there, that at the arterial side we force plasma out. There's a reduction in blood pressure. And then here at the venous side, we call interstitial fluid back into the capillary to replace. And in general, more fluid leaves the capillary than returns. All right, so now we've got the last of the blood vessels to discuss. So now we have turned the corner and are heading back toward the heart. The blood is heading back toward the heart. The blood first travels through venules. These are the smallest of veins. They resemble an enlarged capillary, but as the blood gets further and further away from the capillary network, the venules merge and they start to take on a tunica media. It's always going to be thinner than the tunica media of the arteries and arterioles, but tunica media comes on. We start to see the reemergence of the tunica externa until we get to the veins, which are blood vessels that have all three of these vessel wall layers. So compared to an artery, the veins are thin-walled, and it's all because the tunica media is thinner. So if we're looking here and here at these two blood vessels, which one do you think is going to be a vein and which one is an artery? Hopefully you decided artery here and vein here. And so veins tend to have a larger lumen. The lumen is the interior space that the blood flows in. They just tend to be have larger lumens and be a little more slack in the wall. They don't have near the kind of compressive power that an artery might have. So the blood pressure then in these veins is going to be really low. Remember, as we were leaving the capillary networks, it was only about 16 millimeters mercury. By the time the blood is flowing back up the inferior vena cava, that blood pressure is reduced all the way down to 2 millimeters of mercury. Now this is important because blood will only flow from high blood pressure to low blood pressure. So we've got to have this constant drop across the, along the length of the pipe. But the challenge is with blood pressures as low as 2 millimeters of mercury, gravity has a stronger pull downward than pressure can push upward. So how do we get blood back up to the heart? Well, we've got a couple of cool systems. The first is that there are valves. There we are. These are one-way valves that are found along the veins. And so it's a flap of tissue that folds backward up into the blood vessel, and blood will shouldn't be going in like that. I'm sorry. Blood will come upward like this and make it past the valve, and then if blood pressure drops, then the blood will be pulled downward by gravity, and that closes the valve back up again. And then if we can get another spurt of blood pushing upward, there's another section above here where there's another valve. So we just keep moving the blood from one section of the vein to the next section of the vein, using these one-way valves to prevent the blood from falling or flowing backward. Now, this is problematic, especially for people who do a lot of standing or don't have really good cardiovascular health, because the tissue of the veins around these valves, because it's constantly getting this pressure, back pressure here, can bulge, become larger, and those stretched valves, the stretched valves may develop into varicosities, so varicose veins, and into hemorrhoids. So depending on where the varicosity develops, we call it a hemorrhoid if it's in the rectum, or varicose veins if it's in the large veins of the body. Okay, so there we have it. Everything you ever wanted to know about blood vessels and about capillary exchange.